So we're going to do the first in a couple of videos. I'm assuming that this one is going to take. It's going to be about growth and development. So this is going to be a little bit longer than the other two PowerPoints that I did or the other two presentations that we did for the last group. This one's going to be a little bit longer, and that's because there's a lot more that's involved in growth and development. So I'm just going to talk about this. So whenever we talk about growth and development, there's going to be essentially two different periods. And that's going to be the prenatal period, which is everything that's going to happen once you conceive a child up until the child's birth. And then the postnatal period is everything that occurs after birth. Usually, whenever we're talking about any type of development, we're going to be talking about human development biology. It's just going to be study of how changes occur during the life cycle from your conception to death. Okay? So we're going to talk first. So anytime we're talking about any type of sex cells, we're going to have the first thing we're going to talk about is spermatozoa, which occurs through spermogenesis. They're produced and when we talk about any type of ova, so spermatozoa, which are sperm, are produced by spermogenesis. Ova, which are going to be eggs, are going to be produced through oogenesis. And both of these are essentially just forms of meiosis, in which there's cell division that reduces the number of chromosomes, so that way that you don't have as the full 2N that you would normally have. So normally you have 46 chromosomes, but when you have your sex cells are only going to have 23. And so therefore, that way we can get, mixing and new genes that are going to be passed on from mother to child. Okay? So what happens here is, you remember, you have a diploid situation of your sex cells. Okay? So every time you have a chromosome pair, okay, normally that'd be a single chromosome. However, they've already doubled because that's what they do during DNA replication portion of the waiting phase before meiosis. Okay, so what happens in the first stage of meiosis is we divide, and we get each, instead of dividing the chromosomes in half here, we're going to essentially take one chromosome for each, okay, for each pair, and we're going to put them into two separate cells. So essentially the chromosome pairs separate and go apart. Then in the second phase of meiosis, now we're going to separate out the chromosomes themselves. And so therefore, we're going to have essentially down the middle here of these two chromosomes, or this chromosome, we're going to have one chromosome, part of that chromosome go over here, and another part of that chromosome go over there. Okay? So this is going to be considered what our haploid spermatid is, okay? So that's going to be whatever our sex cells. That'll be our haploid. So they kind of go through essentially mitosis twice. Again, this is just the idea here. Okay? So this happens in spermogenesis. This is what happens. They all look the same and then the same number. However, when you're talking about oogenesis, it's a little bit different. Okay? Essentially what happens is that one of the oocytes the secondary oocyte that occurs is going to be much bigger than the first polar body. So essentially, instead of having an even split that we had here in the first portion of spermogenesis, we're going to have essentially more of like an off-weighted portion that occurs here. And then from there, we have an additional we have additional during meiosis too. Again, these are just going to make more polar bodies. But essentially, we have another situation where we kind of weight more one direction. So instead of like in sperm, we get 4 coming out here. Okay, these are all 4 good sperms. We only get 1 egg, which is again why whenever women go through oogenesis, a lot of times we don't have as many. If usually most people think if there's a fertility issue, it's the woman, and it kind of makes sense because you only get 1 egg per 4 sperms. Okay. So essentially what we have here within meiosis is going to occur with 2 different cell divisions. The first part is where we separate out the chromosome pairs, and then the second part is where you actually separate out the chromosomes. So in meiosis 1, this is where things kind of get a little, I would say, a little screwy, a little bit different. And that's because of the fact that we have this thing where we can have a crossing over. So let's see. Don't see that. Let's see. Okay. So what happens here is essentially we have these two lined up here. But because they're so close to each other, we can have a portion of this bottom portion here of this um, uh, chromosome switches with this chromosome. Does this make sense? Okay. And so that's how the crossing over accentuation occurs. Okay. So it can kind of almost change where genes can be found so you can get more variation. If you're looking for more information on exactly how crossing over occurs, look on page 1092 of your book. It gives a much better picture of what's actually happening there. Okay, better than what I can give you on this PowerPoint. Okay, again, spermatogenesis. The idea here is that we get the same number. It's pretty much the same thing as what we consider for meiosis. Unfortunately, with female sex cells and oogenesis, we get essentially that offset, and so we go from a primary oocyte to a secondary oocyte, and then that eventually becomes the haploid ootid or the ova. Okay. Okay. So again, how do we make the fetus? Well, it's kind of obvious. We've talked about this a little bit. We talked about ovulation. So we go through ovulation, and they'll essentially eject the ovum into the fallopian tube, and then if you have unprotected sex during that time, you have this thing called insemination, where it's basically the seminal fluid from the male is ejected into the female's vagina, and then the sperm travels through the cervix and into the uterus, then into the uterine fallopian tubes. Okay, so going through all those little tubes areas to get to the fallopian tubes and finding that ovum. Okay, so basically we have three different types, essentially stages that occur with fertilization. There's the thermotaxis, and this is going to be when the sperm is attracted to the warmth of the uterine tubes, so it starts going towards the uterine tubes. Then the chemotaxis, which is when the ovum goes and says, hey, I'm going to get that sperm, and becomes trapped in next to each other. And then the acrosome reaction. Essentially what happens there is it allows for the release of enzymes to break down the outer layers of the ovum, and essentially what that allows for you to occur is once that's breaking down, it kind of becomes a blastocyte, which is the fetus, and allows for the essentially fertilization to occur. Okay, so essentially you see here, because it's breaking down the ovum, you're going to get this sperm nucleus to go ahead and get into the ovum's nucleus and match up the chromosomes accordingly. Okay. So again, we want to make sure that we know that we have the plasma membranes and the nucleus enters into the ovum. Essentially, what happens is that these two pairs of chromosomes come together and make the 46 chromosomes that you actually need to have a fertilized zygote. That means that it's genetically complete at that point. It's no longer a haploid, it is a zygote, or it's a diploid. Okay. In addition, you also get the RNA and protein molecules from the sperm. They also enter into the egg, so that way that you don't have to immediately go through cell division. When you go through cell division, obviously for it, there's not going to be a situation of you needing to have different proteins developed right away. Okay, so essentially this continues until birth. So once it goes ahead and is fertilized, it's going to go ahead and implant into the wall of the uterus. Okay, and this is what we call a it's kind of not necessarily happens right away, but once the zygote is formed, it immediately begins to divide. And so essentially it becomes a blastocyte, which is going to be a hollow ball of cells that go essentially develop by the time the embryo reaches the uterus. And then that allows it to implant properly into the uterine lining. Okay, so this is just kind of the showing you how that works. Okay, and this is the early rapid cell division. Again, those are just kind of the early stages that you can see of an embryo. Okay, so all the nutrients, So the ovum has specific nutrients that it's been given until implantation occurs. Okay. Once this happens, once we have this blastocyte that's occurred, and essentially there's 3 different areas. There's the trophoblast, the inner cell mass, and the amniotic sac, which becomes the shock absorbing sac. Okay. So here's just showing you blastocyte. This is going to be your trophoblast and your inner cell mass. Okay. Okay. So once that happens, that amniotic sac essentially is where we start to build our baby, which also gets a placenta. Okay. So this is going to be a structural anchor and nutritive bridge between mother and offspring. So anytime we're talking about anything the mother needs to give to the offspring, it has to go through the placenta. Okay. It also serves kind of as an endocrine organ, but it also works for respiration as well. That's why we talked about, the baby's heart in the earlier on in the semester. It has a little hole in it to prevent blood flow from going back through the lungs because the baby's lungs aren't developed yet. The blood all comes from the placenta. Okay. So we want to make sure that we have the proper fetal blood supply, and it also basically gives you a lot of HCG, which stimulates to continue the secretion of estrogen and progesterone, which is why you're like a hormonal mess when you're pregnant. So the HCG is what you normally don't make because that comes from the placenta. So that is what we use to indicate whether or not you're pregnant or you're not. Okay. So the placenta, this is just kind of the structure in here. So the endometrium is just going to be the area of the uterus. Okay. The uterine wall, you have the umbilical arteries and the umbilical veins and the umbilical cord. This area, this comes out to give you your maternal blood and your fetal blood. And then this, we have the areoles and the fetal areoli. And basically what they do is they allow for the blood to go from the fetus back up to the mother and be processed mother's blood as well. Okay. Okay. So remember, in your first trimester, you're going to have a lot of HCG, which is why it's easy to produce a test to detect that. Obviously, by the time you've reached the second and third trimester, you're showing. So for the most part, hopefully you realize at this point that you're pregnant. But yes, the HCG is what's used in the pregnancy test. Then from there, they stimulate the production of estrogen and progesterone. So those levels increase up until the point that you reach full term and the baby is delivered. I remember that's when we start secreting other hormones. Not to state that you don't continue to secrete those. You do. Because estrogen and progesterone. But the reason why we secrete so much of those is because they're also getting your body ready to have the baby, to be, able to provide for the baby, especially within your breasts. We talked about in the last lecture. Okay, so by the time that you've reached the point of essentially delivery, your one thing that's going to really kind of spike is going to be your cortisol and your prolactin. The other ones are all going to kind of stay the same or jump a little bit, but those are the ones that kind of indicate that it's time to deliver the baby. Okay, so we've got some periods of development here. And we're going to talk about this first one here, and then we'll talk about the rest later. So the first period of development is going to be your gestational period. And again, this occurs during your during the 39 weeks that the baby should be, at least 39 weeks that the baby should be in your belly, still developing. This is what we consider until like almost 3-month segments. They're really not 3-month segments that are called trimesters. So you have your first trimester, second trimester, and third trimester. Now, technically within that is going to be the embryonic phase of development. So up and until that point is just going to be from fertilization to the week 8. Okay, so that can be your second month essentially. And remember, whenever they talk about the periods of development, the 39 weeks, it's not exactly 39 weeks. If you've been pregnant, you know what I mean. But essentially it starts from the time of your last period. So 39 weeks isn't really 39 weeks. And then the fetal phase of development, that extends from week 8 to week 39. Okay, or 40, or wherever you finally actually give birth to the child. Hopefully you don't make it to past 41 weeks. I pity you if you do. Okay, so the first one's going to be the human embryo, and this is going to be more of the fetus. Okay, little cute babies that are in the fetus. Okay, so during prenatal development, in your first trimester, again, they're very, very tiny. A lot of times you can get like birthing trackers that'll tell you like the exact size of your baby. And as you get and again, these are in centimeters. So as you get through your first and second and third trimester, they're gonna basically, and once you make it to term, they're gonna be getting bigger and bigger and bigger, developing more of their things that they need to develop. So specifically like lungs, heart, brain, etc., etc. Okay. So we're going to go ahead and stop here and then pick up with another video on the rest of this later.